In a previous micro lecture, I mentioned how chromosomes are made up of DNA and bits of protein. As you know, chromosomes contain the genes that are passed on to offspring. And in the mitosis micro lecture, we learned how DNA must make copies of itself during the S phase of interphase so that daughter cells receive a full copy of the genetic material. Since DNA makes up most of the chromosome, let's focus our attention on chromosomes. Chromosomes are located in the nucleus of most eukaryotic cells. The numbers of chromosomes in the nucleus vary from species to species. In most human cells, there are 46 chromosomes. In addition, chromosomes exist in the cell at all times. Sometimes the chromosomes exist in the duplicated state, and other times the unduplicated state. So let's talk about these terms for a minute, duplicated and unduplicated. Such simple terms has, in my experience as a teacher, caused lots of confusion and frustration for many students. DNA and chromosomes are really the same thing, and I think this picture shows it pretty well. We see a really great molecular view of the DNA double helix up here at the top part of the picture. But we see the rungs of the ladder, the nitrogenous bases, we see the sugar phosphate backbone and so forth. But if we look at it, take a step back and look at it, like from this view, we see that DNA is wrapped around these pink balls. And these pink balls, what the artist has meant to represent these structures called histones. They're proteins. And because DNA gets wrapped around these balls of protein so much, that's how we can cram so much DNA in a small space. In fact, the statistics I've read over and over and over again, we have about 3 to 6 feet of DNA crammed into every cell of your body. So I think when we look at DNA in this way, we can see how such cramming is possible. Let's look at an individual chromosome. We know that DNA makes copies of itself during DNA replication. You've seen a video on that already. But since DNA and chromosomes are the same thing, let's look at what happens when DNA replicates, but from a chromosome perspective. This top picture here shows us a cell with one chromosome in it. But we know DNA replication occurs here. So if DNA replicates, that also means the chromosome duplicates because of the same thing. So let me ask you a question. If we have one chromosome in this cell, that's one chromosome there, how many chromosomes do we have here? My students in the CETA class, they always get this wrong. How many are there? Well, the answer is 1. We still have one chromosome there. And it drives students crazy. They see 2 structures here where there was 1 before. But that's where the terminology gets kind of crazy. We say this is an unduplicated chromosome. It's unduplicated. But after DNA replication occurs, we call this a duplicated chromosome. So that's not too hard to understand. That's a duplicated chromosome. We say a duplicated chromosome is made up of 2 sister chromatids. So let me maybe outline this chromatid, and here's another chromatid. In effect, we can think of those as being 2 DNA molecules. We had just one at the beginning, but we have 2 here. So a duplicated chromosome is made up of 2 sister chromatids. By the end of the cell cycle, which we'll talk about later, the cell will divide, and each cell gets, some people call these, they get chromatids, and that's technically right, but the terminology to be accurate would be each cell gets a full chromosome. As we see the verbiage on this side, we see chromosomes duplicate inside the nucleus, and eventually the duplicated chromosome splits apart, and each new cell gets its own complete chromosome. So I love this photograph. It's a real chromosome taken probably with an electron microscope. But is that chromosome in the duplicated or unduplicated state? Can you tell? Well, as this big arrow implies, it exists that's existing in the duplicated state. So we can clearly see the 2 chromatids. There's a chromatid, and here's one. And the 2 chromatids are held together with something called a centromere. And it's about where that indentation occurs in this photograph. But let me just circle it there. That's about where the centromere is. In subsequent lectures, we'll understand the importance of that centromere and the importance of your understanding what sister chromatids are. The topic of the next micro-lecture of the cell cycle explains how a cell with duplicated pairs of chromosomes separate the chromatids into individual cells while maintaining homologous pairs in the newly formed cells.